Hello, my name is Egon Willighagen. I work as a researcher at Maastricht University in a group doing systems biology approaches. The title of this presentation, Integrating Lipidomics Data with Pathways, reflects the kind of research that Denise Slenter, PhD candidate in our group, and I have been looking at. While we have not studied this particular dataset, it is a good example of the approach that we're interested in. When we measure experimental data, for example, the perturbation of sphingolipid metabolism in an untargeted metabolomics or untargeted lipidomics approach, when exposed to a compound, as in this example, we might be interested in what actually happens in the cells or in the organism by studying the changes in metabolic pathways, and in this way try to uncover the mechanism of action. Now, these data sets, they typically measure the abundance of metabolites and lipids across a number of experiments, a number of samples, and in each sample this gives you an abundance of a particular metabolite, where when the metabolite is not measured, the abundance may be unknown or zero. So the experimental data that we are starting with is actually after the metabolite or after the lipid identification process. So we already know what the compound is at a certain certainty anyway, and it is this specific compound by means of an identifier or a SMILES that is linked to experimental data across samples. Now, by linking this experimental data, we can actually see which pathways are significantly changed, for which we have, for example, pathway enrichment approaches. But the simplest thing that we can do is actually map the changes in abundances onto those pathways. This we have done a number of years ago for the exposure of or the effects of the exposure of triglytazone, and here depicted on the heme biosynthesis. And what we see in this picture is something that we run into with metabolomics and with lipidomics as well, something that we did not have in the past with transcriptomics. In transcriptomics, you typically measure most of the genes in pathways, but in lipidomics, unfortunately not. And this is very clear in this example because all the gray nodes are nodes without data. Only the blue nodes, those are changes measured in the experiment. Downregulation in this case, that's why they're blue. The green indicates here that the change actually is significant, which may or may not always be the case. So we have a very incomplete story on the heme biosynthesis based on the effect of this compound, and this is what we run into regularly. And there are a number of sources of problems here, and we will see some of them in the next couple of slides. One problem is that we only measure so many metabolites. Fortunately, this is going up, but the number of metabolites that we measure is still low. Here, not lipids. Here, more regular, more traditional metabolites with RS stereochemistry indicated. And when we map this to the pathways, we may not actually have an overlap between the two. We may not measure all metabolites in the pathway, and the pathway may have metabolites that are not even in the sample at all. The latter, for example, when the pathway describes processes that are over multiple cell types, for example. Typical problems. But there are also more technical problems. So here we have an overlap in the tyrosine, but generally a sparseness. The sparseness comes from the amount of data, the amount of pathways that we have, but there is another problem, a problem of identifier mapping. Now, depending on the metabolite of lipid database that you use, the stereochemistry may not be defined. Some databases have a lot of detail regarding charge states, have compound classes, and we will see a number of these situations. The problem here is that these databases have different identifiers. So where our experimental data, each lipid or metabolite has only one identifier in our pathways, this may not always be the same identifier, even though we're looking at the same lipid or metabolite. Now, the sparseness of data in general may not always be a problem. So if pathway enrichment is not really possible, then we still might be able to do approaches like a network approach, as in this example, work from Denise. In the red here, we find metabolites for which there is experimental data, and the green here is too used for to indicate some significance. And again, we see the sparseness here reflected in a lot of white nodes. But we also see the paths between metabolites for which we have data. Now, the idea of this approach is that this pathway, so the intermediate metabolites we may not have measured, too low abundance or too short-lived or something, but following the directed paths, the directed metabolism paths between the metabolites, we might get a richer story of what is going on. Now, for lipids and lipid pathways, of course, we need access to the lipid pathways, and Lipid Maps project has developed quite a number of lipid metabolism pathways with quite a few lipids there with their context, such as which are the enzymes that catalyze those reactions. And this is the recently updated Lipid Maps gateway, and we see a plot on the right side of such a lipid metabolism pathway. We will come back to that. Another aspect of Lipid Maps, of course, is the hierarchy that Lipid Maps provides for all these lipids. What we can see in this diagram here in the middle, we see lipid and a number of subclasses like sphingolipids, sterol lipids, glycerol lipids, etc. And each one of them having further subclasses. Now, this graphical depiction of the hierarchy of the Lipid Maps ontology is actually taken from Wikidata. And the hyperlink in the bottom right, the w.wiki, you can enter that in your browser, and then you get a live version of this hierarchy. Now, Wikidata has a number of hierarchies, and partly derived from information in Wikipedia, for example, but also hierarchies based on the Kebi ontology, for example, which explains why we see a number of messy hierarchical relations, for example, around fatty acid and fatty acid. We will come back to Wikidata and why this is interesting nevertheless later in the presentation. Importantly here is that Wikidata, for this slide particularly, that Wikidata does not just provide a good number of the lipids in Lipid Maps, but also the classes with this hierarchy, and this is something that we can use. Because I already mentioned the identifier mapping problem, and on the left side you see an example of the charge state situation where we have the deprotonated form and the protonated form of one simple metabolite. Each with different identifiers, but we also have compound classes like long-chain fatty acid, which is what we see on the right side. And for long-chain fatty acid, we may still be interested in mapping experimental data for a specific long-chain fatty acid onto this node to actually see what is going on in more detail. But to do so, we need to be able to map an identifier, the data associated with an identifier for a specific long-chain fatty acid to the class of long-chain fatty acid. Now, not for that particular one, but at the top we see one of those paths, pyruvate via one of its conjugates, matching the example on the left, and then to a KEK identifier. So while we do not have a direct mapping of the experimental data with the pathway, we can still link this by taking into account the chemistry of the compound and or the ontological hierarchy of the compound. Now, on the pathway side, our favorite database is Wiki Pathways. It's developed by our group and the group of Alex Pico at the Gladstone Institute in San Francisco. And this is the most recent paper giving some statistics of the database. Already about 2 years old, and what we can see here is the growth in metabolites in the bottom right over there, and also in red the indication of Wikidata. Now Wikidata is going to be important for this identifier mapping, as we will see later. In the bottom left, the Venn diagrams, those give an indication of how much of the human metabolism we already have. Now this was 2 years ago, so the numbers actually right now are even more or better than what we can see here. So I think about maybe 2 years ago or so, we found the lipid maps pathways. We noted that those were images, and we got in contact with the lipid maps team, and in particular with Egon Willighagen. And he actually converted the images of those pathways into interactive pathways for our Wiki Pathways database. And here is one of the examples of that. When I say interactive, I mean you can zoom in on these pathways. You can click the lipids in the pathway, which gives you besides the lipid map identifier, also identifiers for other databases. And even better, on the website, those other databases actually link out to those databases to get more information about those lipids. So based on the initial set of 10 pathways from lipid maps, which by now also have been homology converted to human to pathways with human gene identifiers, we created this lipids portal, of which the lipid map pathways are a significant amount. And besides the interactive display of those pathways, being able to easily link to other databases, another advantage of this Wikidata approach is the curation that we do in Wiki Pathways. So these tags that we have here, the quality tags, it doesn't just say that they have been curated with lipid maps, but there is also a number of automated tests that are run on top of the pathways. And here we see some of the tags. So at that moment, this pathway did not have a description yet. There were some unconnected links, which particularly matters if you want to do some computing based on this data. But we can also do additional tests in an automated way, which these tests are being run every night. And in the bottom right, we see, for example, an automated test for secondary CABI identifiers. These secondary CABI identifiers are older CABI identifiers, and we now actually get a signal when one of those older CABI identifiers is used in one of the pathways. So that we can update them to the most recent one. For each pathway, there are about 70 automated tests being run every day on them. Now, the drawing of these pathways, there we use PathVisio. And PathVisio has been around for some 12 years now, and the advantage of PathVisio for drawing these pathways is that it allows us to express in a machine-readable way quite a bit of information. I already mentioned the identifiers, which we see on the right side here of the PathVisio window, and in the middle we see the one particular enzymatic transformation with a literature reference, a small one on myristic acid, some metadata at a pathway level, and the semantic meaning of this enzymatic reaction. What must be clear is this identifying mapping, and for that we actually really needed a solution as well. Most of the pathways use a variety of identifiers from a number of databases, and while this has been standardized to some level, we still see in last year quite a diversity of identifiers being used: CABI, HMDB, CAC, CAS, but also 148 lipid maps identifiers. Being able to convert them when analyzing data is essential, and the tool we use for that is BridgeDB, which you can find on bridgedb.org. Now, the source of these identifiers have been traditionally CABI and HMDB, but we realized that a number of these identifiers were not available, and lipid maps was one of them, and we started using Wikidata for that. But Wikidata and Scolia actually has a number of additional advantages. I already mentioned the hierarchy, and on the left side for fatty acid. It also has links to the English Wikipedia, some redirecting, and at the bottom you can see actually the redirect in action for a lipid maps identifier. And by using this approach, Wikidata and Scolia actually provides us with additional information, crowdsourced, just like with Wikipedia itself, about chemical compounds. So physical chemical properties, and very interestingly, publications that mention or describe that chemical compound. So in summary, what this presentation has shown is that we can map experimental data to biological process diagrams. We typically have sparse data, though we have network analysis approaches to overcome that, to handle the sparse data as well. And we saw the importance of the identifier mapping, even if we have the chemical identity determined, and we haven't even gone into uncertainty in that molecular identification. We also saw Wikidata and Scolia for a linked data approach, providing us with a lot more information on lipids and ways to keep updated on a specific lipid. We saw Wiki Pathways as our pathway database and a couple of the tools that we use for this to make all this happen. With that, I end my presentation. I would like to thank the people in our group, and of course the projects that funded the work on Wiki Pathways in general, and the tools around that, as indicated up there, and of course very prominently Lipid Maps for providing us with a number of metabolite pathways to the Wiki Pathways database. Thank you everyone, and I'm open for questions. Thank you.